Hello and a very warm welcome or a welcome back to this second session of our ISNTD Climate and Health Conference 2023. Over the course of a day, an event where we are trying to get a better understanding of the linkages between climate and health, but through the lens of infectious diseases and particularly neglected tropical diseases. We just had a really rich, packed, and very informative first session looking at data and modeling for climate and health linkages. We've heard as well of the stark urgency with which we need to understand and tackle the impact of climate on health very rapidly, an impact that has the potential to reverse decades of progress that have been made in the health, particularly of the most vulnerable communities. So really an important moment this year to understand the evidence base and to see exactly what's happening when it comes to the impact of climate on diseases. But at the same time, despite a huge amount of collaborations and partnerships building, still lots of gaps remaining, whether this be in the data or in some of the research areas. And so in this session, we're moving away from the modeling aspect, and we're really going to be focusing at some of the evidence base. What has been happening across a number of tropical diseases in terms of the climatic variables and their impact on diseases? We'll be hearing examples from diseases as varied as polycystic echinococcosis, yellow fever, but also very interestingly, impact of temperature and rainfall on snakebite. So 3 very different approaches, which will help us to understand a little bit more about these all-important linkages. And before we start on all that, we'll have a wonderful overview of what are the research trends, where are we going, but also all importantly, what's been missing. So a really exciting session ahead of us. It's my absolute pleasure to welcome our 4 panelists this morning. And first we will hear from Paige van der Vuurst. Welcome, Paige.
Thank you so much for having me.
Paige, you're joining us from the Virginia Polytechnic Institute and State University, where you are doing a PhD in the Laboratory of Disease Ecology. So a very warm welcome to you.
Thank you so much.
We will then hear from Dr. Adrià San José Plana. Hello, Adrià. Adrià,
Hello.
you're Predoctoral Fellow at the Barcelona Institute for Global Health, or ISGlobal, and having had a whole session with the Barcelona Supercomputing Center, I'm starting to see a trend here between Barcelona and climate and health work. So it's lovely to have you here. Our next speaker will be Annabelle Monet. Hello, Annabelle.
Hello, Marianne. Nice to meet you all.
Nice to have you here. Annabelle, you are the public affairs lead, global public affairs lead in vaccines at Sanofi, and you'll be talking to us about yellow fever and climate change. And finally, joining us from Colombia, Dr. Carlos Andres Bravo Vega. Welcome, Carlos.
Hello, Marianne. Hello, everybody. Thank you so much for the invitation.
Thank you, Carlos. You are with the BioMac investigation group in computational and mathematical biology at the Department of Biomedical Engineering at the University of the Andes in Colombia. So welcome to the 4 of you. And now, without any further delay, I hand over to you, Paige, for your introduction to research trends and also research gaps in the linkages between climate and disease.
Thank you all so much for having me. Apologies if it's a little bit dark here. I am in the United States, so it's rather early. But today I'm going to be talking to you about climate change and infectious diseases, reviewing evidences and research trends across the last 5 years. Again, my name is Paige van der Vuurst, and I'm a PhD student at Virginia Tech. So climate change and infectious diseases, we know, are 2 phenomenon that have massive impacts across all major geographic areas and across almost all biological systems, from severe weather patterns causing more fatalities and injuries to impacts to water availability and quality. We know that climate change can have massive impacts to human health. We also know that the life cycle and propagation of many pathogens are intimately linked to climate. We also know that humans do not exist inside of a vacuum. We interact with hundreds of different species every day, and we know that when climate impacts where and when different animal species live, it can impact the pathogens that they carry as well. So host distributions can be altered by climate change. We also know that the global burden of infectious diseases is not distributed equally across the globe. If you look at this map where darker colors indicate more infectious disease burden, you're going to see a trend of countries in tropical or subtropical environments, countries that have more limited public health infrastructure, or countries that experience persistent geopolitical conflict are bearing a higher burden of infectious disease than more well-developed countries in temperate areas. We also know that climate change impacts are not distributed equally across the globe. Again, here showing darker colors signifying higher climate change impacts, countries that have higher human densities, countries, and again, those tropical and subtropical ecosystems bear the brunt of climate change impacts. And in terms of neglected tropical diseases, we know, or many of you know, that neglected tropical diseases span a variety of different pathogen types, from viruses to helminths. So it's important for us to understand how climate change impacts all diseases and what research has been done on these specific types of pathogens. And if we look at a map of people requiring interventions against neglected tropical diseases, we'll again see that there's a trend of some countries experiencing a higher burden than others. Again, some of those same areas that are at high risk for infectious disease impacts and climate change impacts are also the ones that are bearing the heaviest burden for neglected tropical diseases. So what we wanted to do in order to understand what research has been done on climate change and infectious disease and what the current trends are in that field, we wanted to conduct a systematic scoping study, which allows you to look at the current landscape of research, identify research questions, current trends in research, and also collate, summarize, and visualize data from often disparate fields that are sometimes not connected. And this also gives us a roadmap forward so we can understand what we have done, what we haven't done, and where we need to go in the future. So that's exactly what we did. I and my co-authors gathered every piece of scientific literature that we could from the year 2015 to 2020, so the most recent pre-pandemic period of research, from all available data sources such as Clairvoyant, university libraries, PubMed, Web of Science, and we looked to see what the current trends are in that field. However, after we looked at the initial over 10,000 publications that we got during this period, what we found is that only about 621 of them actually fit our inclusion criteria and assessed climate change impacts on infectious diseases in an empirical manner. So what this told us was that there's a lot of literature using climate change as a keyword, and they are maybe not exactly assessing climate change, but maybe climatic variables like temperature or precipitation, but not how that variable has changed across time. We then extracted the data both on the diseases being studied and the individuals doing the research to assess both study and authorship trends. We then collated and assessed the trends that were present. So one of the big results from this study was that over half, or 59.6% of the articles studied during this period reported that climate change was having a definitive impact on the disease system that was being assessed. Only 8% of the papers found that there was no impact of climate change on the disease system that they were studying. Something interesting that we found, though, was that about 33% of the papers reported a possible or potential impact of climate change, but they could not say one way or the other if there was a definitive impact or not. So looking at those papers that did report a climate change impact of some kind, we found that 81% of them reported that climate change was increasing the burden of the disease system they were studying. Now, This could be an increase in the total suitable area for that pathogen. It could be an increase in vector capacity. It could be an increase in the propagation rate of a vector. It's somehow making the system worse or the situation worse. There was 11% of the papers, however, that identified that climate change was actually going to decrease the burden of that infectious disease. Now, this could be an example where areas in Africa will become too hot for the plasmodium that causes malaria to continue to exist inside of some vectors. And then again, we found 8% of the papers that reported no impact of climate change on the disease system. So what this means for neglected tropical diseases, which many of you study, is that based on this subset, we are likely going to see an impact of climate change on the disease systems that you study. And based on this subset, it's very likely that that disease system could be augmented or increased in burden based on this subset. We then wanted to look at where research was occurring to see if we could ascertain if there's an equitable distribution of research across space. And when we looked at the areas that had more research conducted in them, we noted that countries that had higher populations had more research, which makes sense. More people, more researchers. However, when we corrected for population and we looked at research effort per capita, a more obvious and slightly concerning trend appeared, where you can see that countries like Canada, Norway, and Australia, areas that are far away from the equator in more temperate regions, were receiving a disproportionate amount of the research effort. However, countries in tropical or subtropical areas, the ones that we know are at greater risk, were not as well assessed. So again, if we look at this map of emerging infectious disease risk, you'll see that these countries that have higher human densities in tropical areas with higher mammalian biodiversities, areas that have persistent landscape change, as well as climate change impacts, are at greater risk. So if we kind of toggle back and forth between this map of research effort and the map of risk, you'll see that these areas are not exactly overlapping in the way that we would like them to. We looked at the exact animal taxa that were being studied during this period as well, and overwhelmingly it was humans, which makes sense. Human health drives many research efforts. However, there was also a very large emphasis on arthropods. However, other vectors or organisms were not as well assessed during this period. We then looked at the specific transmission types being assessed during this period, and overwhelmingly it was vector-borne diseases followed by food and waterborne pathogens. However, other types of transmission, more directly transmitted diseases, not as well represented. And then when we looked at the specific diseases being assessed, there were over 100 different diseases that were assessed during this period. Not all of those are listed here. This is only the top 20 or so. But again, things like malaria, dengue, Lyme disease, Zika, chikungunya, those vector-borne, specifically mosquito-borne pathogens were very well assessed during this period, followed by things like diarrheal diseases or cholera. However, again, more directly transmitted pathogens or wildlife pathogens not as well represented. Looking at specific vectors, again, mosquitoes were very well assessed during this period, followed by ticks. However, things like kissing bugs or mites were not as well assessed. Very few publications looking at those, the impact of climate change on those vectors. So why exactly was there such an overwhelming assessment or emphasis on vector-borne diseases during this period? Well, we know that vector-borne diseases account for more than 17% of all infectious diseases, causing more than 700,000 deaths annually. We also know that vector-borne diseases are the fourth leading cause of death for children under the age of 5. We also know that vector-borne diseases have heavy economic impacts, especially to countries that bear the brunt of the burden of infectious diseases, not just in morbidity and mortality that occurs in those areas, but also in preventative measures that countries put into place to try to reduce the burden of infectious diseases. We also know that there are clear links between climate and vector prevalence. It's very easy to understand how factors like temperature, precipitation, humidity impact vector capacity or vector prevalence, because things like mosquitoes can be studied in both laboratory and field settings. However, when you're looking at how things like temperature and precipitation impact more complex vectors like birds or mammals, there are many lurking variables that can be present. So it's a little bit more difficult to draw the links between climate and the prevalence of those kinds of vectors. We also wanted to look at authorship trends, so not just where research was being conducted, but who was doing that research. And again, here, darker colors are signifying more authors from those areas. And these blue dots represent the institutions that were most published during this period. And so those were in countries like China, the United States, and Australia. So again, what we found is that authors generally tended to do research in the countries that they were affiliated with a couple of notable exceptions, such as countries in the Middle East and Mongolia. We're mostly seeing that people are doing research in their own backyards, so far less parachute science than we were anticipating. We also wanted to look at authorship trends in terms of gender expression. We know that women have been historically underrepresented in many STEM fields, so we wanted to assess if that gap between authorship was still present. And what we found was that there was more male or masculine pronoun use in first authors. But when we looked at senior authorship pronoun usage, we found that that gap between he, him users and she, her users was much greater in senior authorship positions than it was in first authorship positions. Now, what this tells us is that there is still a disparity between authorship in terms of gender expression in the field of climate change and infectious disease research. However, that gap could potentially be closing. We also looked at the institutions conducting research, and overwhelmingly that was colleges and universities, so those secondary education institutions, followed by governmental bodies. This was somewhat surprising to us. We expected there to be a greater representation of governmental bodies doing research on climate change and infectious diseases, due to the fact that many governmental bodies have to deal with the impacts of climate change on the public. However, there was, again, more representation of colleges and universities. So what exactly did we find kind of in summary? Well, we know that geographic biases and both subject and taxonomic biases are present within the field of climate change and infectious disease research. More research was occurring in temperate, well-developed areas, and more research was being conducted on mammals and arthropods during this period. Now, when biases are present within a field, this inevitably leads to neglect, as many of you are aware. And some neglected areas that we identified were developing countries, countries in tropical or subtropical areas, and countries that experience persistent geopolitical conflict were sometimes not well studied during this period or not studied at all during this period. So conclusively, we also identified some neglected disease systems during this period that were not well studied. Some of those are, again, those neglected tropical diseases that we are all so aware of here. These include parasitic infections, directly transmitted diseases such as rabies, which is something that I study very closely, wildlife-borne pathogens, which is concerning because we know that many wildlife pathogens can spill over to livestock or to humans, causing disease emergence, which can lead to pandemics. And also upper respiratory infections were not well assessed during this period. However, there's one massive caveat there in that all of this data was collected prior to the COVID-19 pandemic, or the largest impact of the COVID-19 pandemic. So if you were to assess climate change impacts on infectious disease research after the year 2020, I guarantee you there would be a larger emphasis on upper respiratory infections such as COVID-19. So definitively, there are some limitations and recommendations that we gleaned from this study. First was that the English language required for publication could be a barrier, especially for some of the countries that we identified as being underrepresented. We also wanted to point out that author interpretations of results could be biased or could be shaped by publication pressures. So what we found was that there was a large proportion of research that had statistically significant positive results associated with climate change and the disease system. Now, this could be because there is a reticence to publish null or negative results within the field. However, if we are not publishing those null or negative results, we're not getting a full picture of the current situation of climate change impacts on disease systems. So we want to be aware of that as a scientific field. We also wanted to call for an increase in collaboration, especially with countries that we identified as being underrepresented in this field, especially for those of us that do come from those well-studied, highly developed, higher GDP countries. What we know is that when authors from underrepresented areas collaborate with or publish with authors from more well-developed temperate areas, their publication rates go up. So as we work to make publication and literature more equitable for people, it's important for us to collaborate with those groups so that their research can be put out there. We also wanted to call for a more ecological model of health study rather than an anthropocentric one. Now, I know that that's somewhat counterintuitive because we're all here to assess and talk about neglected tropical diseases that impact humans. However, when we don't study the ecological impacts of different vectors and wildlife species, we're missing part of the picture of potential spillover dynamics that could lead to the next pandemic. So if we're so overwhelmingly focused on humans that we miss the impacts of climate change on wildlife reservoirs, we're going to be left wrong-footed when the inevitable next spillover occurs. And with that, I would very much like to thank all of my collaborators at the Laboratory of Disease Ecology and Biogeography, my advisor, Dr. Luis Escobar, all of the other graduate students in my lab, and our undergraduate researchers, the Virginia Tech College of Natural Resources and Environment, as well as the Translational Biology, Medicine, and Health program, and our funding agencies. And just as a disclaimer, the opinions expressed here are those of me and my co-authors, not necessarily those of our funding agencies. And with that, I will take any questions, or if we want to save questions till the end, that's totally fine.
Paige, thank you so very much for your presentation. So many things in just 10 minutes that I think the field of infectious diseases, of neglected tropical diseases is very aware of, but to see it really organized and outlined in such a clear, useful and concise manner, I think is really helpful, particularly not just focusing on gaps by disease, but things such as the English language, and also really reminding everyone of this One Health approach. So thank you. That's been an incredibly useful tool to shape any future research, and also take a moment to take stock of where we are as a field and where we need to go. So that's been really wonderful. We'll now turn to our three speakers who are going to be focusing on particular neglected tropical diseases and give us a bit more insight. First of all, I'd like to invite a presentation. So Dr. Adrià San José Plana, in collaboration with Dr. Javier Rodo, both at ISGlobal, and we'll be hearing more about how climate determines transmission hotspots of polycystic echinococcosis, with a focus across Pan-Amazonia. Adrià, over to you. Thank you very much.
So, well, thank you for having me here, and nice to meet you all. My name is Adrià San José, and I'm going to be presenting some work we've been doing regarding polycystic echinococcosis in Pan-Amazonia. And this is already published in a paper in PNAS. Here you have the link in case you want to take a look into that. It's going to be obviously better explained than what I'm going to be able to do here in 15 minutes and so on. But let's start. Polycystic echinococcosis is it's a neglected zoonotic disease. It's a parasitic disease. It's called by a cestoad called Echinococcus bogelli. This disease is potentially deadly among humans. It has a case fatality rate quite high of around 30%. And it naturally circulates in Pan-Amazonia thanks to a two-host sylvatic cycle where wild dogs can act as final host, and some rodents, especially the Cunuculus paca, can act as intermediate host, and thus allowing the parasite to complete its life cycle. The thing here is that the hunting of the Cunuculus paca, and the paca is a very appreciated and heavily hunted animal, allows this disease to sort of establish a domestic transmission cycle where domestic dogs can act as final hosts and substitute wild dogs, and humans can act as intermediate hosts, then allowing again the full cycle of the parasite. As I was telling you, this is a neglected tropical diseases, but there are different sort of also neglected tropical diseases, and this is one of the most truly neglected ones. There are only around 200 human reported cases in history, which is the evidence that we have here. Obviously, it circulates silently, but these are the reported cases. Here you can find the evidence that we have in these purple points of human infections, and we also have evidence of infections in animals, especially in pacas, that you can find here in blue. Obviously, well, needless to say, this is Pan-Amazonia with all the different countries of the region. And this data is overlaid on the presence of this intermediate and final host. This is very rich data that will help us understand this disease better. So, what are the objectives of our research here? Basically, to answer two open questions. The first one is, which is the spatial distribution of the disease? This is still an open question. As I was telling you, this is a truly neglected disease. And second, in a more maybe scientific fashion, to try to understand which factors determine environmental factors, climatic factors, cultural factors. No, what of these factors can help shape the spatial distribution of the disease? And here we'll pay attention because we will separate between the sylvatic circulation of the disease, so how it naturally circulates in Pan-Amazonia, to the spillover of the disease, so where it jumps to humans. And finally, obviously, we will be able to answer the final question, which is whether this is a climate-modulated, climate-driven disease, and if this is the case, which is the mechanism by which this takes place. So what we did is to run two separate models, as I was telling you before, and given that our data, or let's say we have data of human infections and of animal infections separately, we decided to run two different models, an animal model, animal infection model, and a human infection model, to see how different covariates affect the process, like different stages of the transmission process. Temporal information here was very scarce, so we couldn't approach this with typical time series or approaches or other time-aware statistical methods. So we had to move to spatial models, and especially we chose maximum entropy models for a series of reasons, and two of them are that they only require presence data, so where this disease is present and not absent, which is also hard to establish. And secondly, because they provide metrics or indicators of the variable contribution and response functions. Basically, this means they allow a sort of readability or understanding of the process, which was key for us at this stage. Which are the factors, which are the covariates that we think can influence the disease? Obviously, first of all, the presence of the host, the presence of intermediate hosts, and the presence of final hosts. Third, obviously, the distribution of the human population. We're talking about also human infections. Four, and this is a bit harder to assess, to try to know which is the hunting interest and the hunting pressure for pacas, as this is a key aspect for the disease to jump to humans. And finally, we wanted to explore whether land use change, and climate change could impact how this disease circulates. However, this was more of a wish list of things that we would like to have, not things that are there available for us to use, because only number 3 and number 5 are there available for anyone to use. So before we went to the modeling part of the Maxent models parts, we had to do a lot of previous work. So first, what we did is we applied ecological niche models to try to derive the first two covariates, regarding the presence of hosts. For number 4, for the hunting interest in pacas, we used a massive hunting data set that our local co-authors had assembled throughout the past years, and used kriging, so sort of a statistical interpolation, to derive the sort of continuum estimate of the hunting pressure for pacas. And finally, regarding climate change impacts, we use an advanced time series decomposition tool called singular spectrum analysis to derive ourselves some indicators of climate change. I'm not gonna dwell on this because we don't have time, but this is just the same, like how we derived this covariate. This is more like of a graphical summary. Methodologically interesting, so we can talk about this later at some other point if someone wants, but now I'm gonna skip it and go to the next part. Just let me say, and let me focus on this 6 covariate, the climate change impacts. What we did, we took the time series of the last 30 years of, for example, temperature, and we made a decomposition of this time series on trends, of how it has been changing. And then also in interannual variability, which in Panamazonia is seen to be ENSO-driven. ENSO meaning El Niño Southern Oscillation determines this interannual variation of temperature in Panamazonia, and this ENSO has two main modes of variability, which are the quasi-biennial and quasi-quadriennial modes of variability that have different spatial patterns, so we decompose them as well. So for each variable, like for temperature, we derive 3 indicators, trend, QQ, and QB, and also for humidity or precipitation and so on. So finally, everything was ready to run the max-n models. We had all our covariates, the one we retrieved, the one we built, the training data, and so on. So we ran the 2 separate models. We did this in a rigorous way. We looked over all the key parameters in max-n's, regularization terms, features, and so on. We did an assessment of the skill of the model with a cross-validation. And given that the skill was similar between some of the models, we analyzed the 5 top-ranked models basically all together to make sure we had sound, stable results. So here we have the results of the first model, the animal model. And if we're short of time, let me just focus on panel B. We have the permutation importance. This means how important is each variable in accounting for the spatial distribution of the disease. And let me just focus on the most important variable, which is this QBT, this quasi-biennial mode of temperature, which if we go to panel C, you will see it has a response function which shows a decreasing trend, meaning that a higher QB translates into a lower risk of having sylvatic circulation of the disease, and on the contrary, a low QB means a higher probability of having the sylvatic circulation of the disease. Then we had other functions, land use, natural suitability, which basically account for the common presence of intermediate and final host and so on, but I don't have time to comment on everything here. For the spillover model, results were different. We were expecting this, obviously. The most important variable was the distribution of population, which makes complete sense. Humans are the ones that are getting infected. But secondly, the first environmental, let's say, variable was the quasi-quadriennial mode of temperature, like the other one, not the QB like before, now the QQ. And if we look at the response function in panel C, this is the second one here, you see that what we find is that the higher the QQ, the higher it is the probability to have spillover, basically, of the disease. Apart from population and QQ, then we follow with land use, the QBT, and these are the exact functions that we saw before with same response functions, and so on. Well, so given that these 2 variables, this QB and QQ, stood out as very relevant variables, we took some time to try to better understand them. So here you have their spatial patterns. The QB here on the left presents a sort of dipole. We see, like a hotspot around the Guyana shield, and the other dipole maybe being the Western Brazil or also Peru, Bolivia, and so on. Whereas the QQ has a very different shape, and as you can see here. If we take a look at some of the decompositions of temperature, and panel A here, we are focusing in a point in the Guyana shield, we see that indeed temperature shows this very marked, strong biennial cycle. So every 2 years, temperature goes up, and so on. And this is why here we see a hotspot of this QB, and here the QQ, which is in red here, is not so important in explaining temperature. Interestingly, if you look at the graph below, you will see that these like pulses, these biennial pulses of temperature generate rainfall afterwards in a convective manner, okay? If we now move to panel B, which is like in the other dipole in the southwestern Brazil, what you see is the temperature there is much more stable. There is not this QB cycle, and QQ basically dominates. But the reading also with precipitation is that everything is much more stable, especially if you look at the axis, you will see that the ranges are lower. So then we went on to try to understand how the QQ that only appears in the spillover model, so not in the natural circulation of the disease, can affect the spillover process, and why and where exactly. So we went back to the amazing hunting database that I told you before that our co-authors had assembled during the last 30 years, and we constructed an aggregated time series of hunting practices in Panamazonia that you can see here, and we derived a sort of low frequency time series. And we did the same with the ENSO, the El Nino, as I was telling you. This is what is forcing the climate in Panamazonia, and this is basically the same as the QQ, okay? So we did a sort of analysis on how one of if the ENSO affects the other variable, the hunting variable, and we found very strong associations, statistical associations of how El Nino was affecting hunting practices in Panamazonia. And they were anti-correlated. Throughout the whole period, there was just one anomaly here in 2010. You can see here it in red, but this can be explained because this was a weird year in terms of the Nino, a Madoki type, well, it doesn't matter. But we can also explain this and therefore see that there is a strong anti-correlation between the two variables. So now we go to the last part, which are the mechanisms, the most interesting one. What have we found? Our results regarding the QB, they show that we find the same response function showing up in both models. And we read that as a transfer of information from the sylvatic model to the spillover model. This is natural because for humans to be infected, we need the disease to be there. We need sylvatic circulation in the same places. The way we read that is that Ebogeli thrives in the areas with less QB amplitude. This means in places which are more stable, at least stable in terms of temperature and precipitation. And we just read this as a sign of how this parasite thrives in stable environments. Why is this important? Probably these variables can have a direct effect on the parasite when it's waiting to be ingested and so on the floor. Proper levels of humidity are essential, and excessive humidity or droughts can kill this parasite. And we can have also effects on the host, on their behavior, populations, and so on. Regarding the QQ, this is a variable which was the most important environmental variable in the spillover model, and it was only showing there and not in the other model. What we found is that the spillover risk of Ebogeli increases along increasing QQ amplitude. Our co-authors were always telling us that the Amazon experienced seasonal changes in food sources. Fishing being dominant during dry season because water is low and their fish accumulate and it's easy to fish, and hunting then becoming more prevalent during humid season, because there's too much water, it's hard to fish, and people switches to hunting. What we found, we think, are the same differences, but in an interannual scale. When we have a warm ENSO event, an El Nino event, this leads to drier conditions in Panamazonia, especially in the south, and this favors fishing as the main food source. This results in a decrease in hunting, and if we have a decrease in hunting, we have a decrease in the spillover risk of Ebogeli to human. And we see exactly the contrary during La Niña events. The interesting thing here is that the max N spillover model identified the ENSO cycles, the QQ, as the great, which basically tells us of the places where we see a greatest dry-wet differences in intraannual variability, as those were hotspots of Ebogeli transmission. And our time series approach then showed us that probably what we see is ENSO-driven hunting peaks that contribute to outbreaks of Ebogeli spillover. So this is my final slide, the conclusions of the paper. The first one, the most basic one, is that we derived the first detailed maps of polycystic echinococcosis, and we derived them separately for the sylvatic circulation of the disease and for the spillover to human. A very interesting result was that the human model incorporated the animal model results. It was trained with completely different data and so on, but this is something natural because for humans to get infected, we need sylvatic circulation of the disease. So it's natural that some of the information is naturally transmitted to the human model. Our key and most important result is that polycystic echinococcosis is a climate modulated disease. And we show that how and why. First, we show that PE's, polycystic echinococcosis, sylvatic circulation is favored by climate stability, let's say temperature and precipitation, to what we could see. But then we also saw that the spillover risk is affected by these ENSO-driven disruptions of hunting practices. The interesting thing is that results are generalizable here to other zoonotic diseases which are associated with hunting practices in Panamazonia because we identified the concrete mechanism. If you want to put this in a broader context, it's important to recall that ENSO extreme events are intensifying, and they are projected to intensify even more due to global warming. Therefore, we can infer that climate change is indirectly driving the spillover of Ebogeli, How? By amplifying ENSO variability. Okay. And finally, well, this was quite a methodologically challenging paper because of the nature of our data. Analysis on the temporal domain, at least of infection data, was initially impossible. But given that some of the covariates had temporal information, the climate, the hunting, we could explore some of these patterns, identify some of the potential mechanisms, and complement the static results provided by the MaxN. And well, I leave it here. I'm sorry if I went a bit too fast, but thank you for everything.
Adrià, not at all. That was amazing. Thank you so much. What can I say? It's been very impressive to see that despite the many challenges we've been talking about in this session, but also earlier today, and just more generally when bringing together climate and disease studies and research, that you've been able to overcome so many different of the challenges, from the data to the mapping and so forth. And then with this fantastic result of creating not just the first detailed maps, but also really being among some of the first research linking echinococcosis and seasonal or climatic impact. And with echinococcosis being really very neglected, even amongst the neglected diseases, and with lots of applications all the way to the Mediterranean region. So it's been a wonderful presentation. Thank you so much for bringing that insight to this panel. And so now we'll move on to another disease, which is yellow fever. And I'd like at this point to invite our next speaker, Annabelle Monet. Thank you for joining us. I'm going to hand over to you. You are Global Public Affairs Lead in Vaccines at Sanofi. And what's really interesting is that yellow fever being one of the vector-borne diseases, this is really some of the attention that really dominates the linkages between climate and diseases. But it'd be very interesting to hear what are the drivers behind the expansion, what are the actual linkages between climate and some of these diseases, and why should they really be placed right back at the top of the public health agenda. Annabelle, it's over to you. I'll put up your slides.
So first, thanks, Marianne, for this full day dedicated to this key area of concern. And I think even more than ever, I think it's important on this topic to foster exchanges and data on science, create awareness and collaboration also. And you will see that it's true for neglected disease, and it's true also for some old disease supposed to be well known, as I will come on it. So before going into the details, just a brief disclaimer to mention that source, illustrations, references are from a visual story that was on yellow fever that was published by Devex in April this year, and that we sponsored, and you can find it easily on the web, just typing yellow fever visual story and Devex. So going now into the story, I think we all remember how far COVID impacted the world, impacted our own life. But long before COVID, a strange disease really swept across the planet, spreading to the Americas in the 15th and 16th centuries, and believed to be originated from the slave ships coming from Africa. And from there, many outbreaks occurred in the North America and also in the harbor in Europe. So in the 18th and 19th century. And as an example, in Philadelphia, for example, there were an outbreak with more than 5,000 deaths, so around 10% of the population. And also in Europe, in Barcelona, I know we have lots of people from Barcelona today, there was a large outbreak with almost 20,000 deaths. So 1/6, I think, of the population of the city at that time. So really quite a large outbreak. And also the disease influenced also a lot the history. For example, the disease decimated the troops of Napoleon in Saint-Domingue, and finally it makes him really give up his plans to the Americas. So really a fascinating history of this disease, and at that time it creates really a lot of mystery. What is this strange deadly disease with no cure, creating so impressive outbreaks? And again, yeah, the research around the disease is really fascinating for all the researchers that are online. And for example, there were some doctors that were lying during 7 days with the clothes and in the bed of sick people that were dead from the disease to try to understand. And so they concluded that it was non-contagious disease. And there were lots of studies to try to understand how it was transmitted. And we needed to wait for Carlos Findlay's hypothesis and also Walter Reed's research at the end of the 19th century and beginning of the 20th century to identify that this disease is transmitted by mosquitoes. So yellow fever, as I said, is a mosquito-borne viral disease. Initially, it was called black vomit. The symptoms include fever and nausea, but in extreme case, in fact, it can induce hemorrhage or bleed from the eyes, nose, and mouth. And the name yellow fever is coming from the jaundice and the yellowing of the eyes. The disease is mostly asymptomatic, but for the small proportion of yellow fever patients that develop these severe symptoms, half of them will die within 7 to 10 days. As I said, it's transmitted by the bites of infected mosquitoes, mainly from Aedes and Echinococcosis species. And importantly also, this disease is a zoonose, so it's carried by primates, both humans but also non-humans, so monkeys, with 3 types of transmission cycles. A jungle or sylvatic cycle between monkeys in the forest, an intermediate cycle between monkeys and humans, and also urban cycle between humans only. And I think you will understand there why we're making the link between climate and this disease today. But before that, where are we today with yellow fever? So thanks to all the research findings on the vector and also on the viral nature of the disease, drastic mosquito control measures were put in place along with vaccination, because vaccine is available since the 1930s, and really all that helped to pose yellow fever's reign of terror that I was describing just before. However, yellow fever still circulates in several low and middle income countries. As of November 2022, 42 countries and regions were still at risk of the disease. And we estimate that 109,000 severe cases and 51,000 deaths each year, 90% in Africa. And we do think that these figures are really, really underestimated. Importantly for today's topic, what we observe is a re-emergence of the disease in the past 6 years with explosive outbreaks, including in areas where there were some preventive mass vaccination campaigns in the past. And this re-emergence is fueled by insufficient vaccination coverage, population growth, population movement, urbanization, but also climate factors, and let's deep dive in these climate factors. So as pretty well underlined by Paige, climate change is well known as a key driver for the ecology of mosquito vectors. Thinking about warmer temperatures, it will really help the development, the growth of vector, and even makes the reproduction cycle of the mosquitoes and also of the virus more fast. thinking about the rainfalls that really favor the breeding sites for the eggs of the mosquito, and then can really increase the abundance of the mosquito vectors. So all that can really exacerbate the disease in the endemic area, but most importantly, it can make the vector expand to new areas where people are not yet exposed to the disease or not yet vaccinated. And this could be dramatic because we could maybe observe again what I described in the past when ships from Africa were coming to areas where the population was not exposed or not vaccinated yet, and induced quite large outbreaks, urban outbreaks. So the models predict that by 2050, nearly 500 million more people would be at risk for contracting mosquito-borne diseases like yellow fever, dengue, chikungunya, and Zika that are transmitted by the same kind of mosquitoes. It predicts also yellow fever deaths across Africa that could increase by up to 25% in the worst case scenario. And last but not least, yellow fever mosquito vector could reach new temperate areas in the US and China, as far north as Chicago in the US and Shanghai in China. So again, it could be really dramatic, knowing the lethality of the disease during outbreaks in a urban area where the population is not yet immunized, either naturally or by vaccination, through vaccination, sorry. Of note, it's not just autumn weather, but also human behavior change that could expand the disease, that can cause, the expansion of the disease. Thinking about extreme weather events, it can cause social changes like mass displacement, causing thousands of climate refugees relocated from rural communities to major cities, and that could induce large urban outbreaks. Another environmental issue linked really to the nature of the disease itself, so the zoonose, is the deforestation. Because in that case, people will go more often in the forest to work, we live closer also to the forest, and so will be more at risk to be bitten by an infected mosquitoes infected by monkeys. So all that could really exacerbate the threats of yellow fever, along, as usual, with insufficient vaccination coverage. So there is, despite hard to predict variables, there is overall a consensus among experts that changes in weather and migration patterns will contribute to higher burden of yellow fever in the future. So what should be done to get prepared to address this expected rising risk of yellow fever? Several strategies could be put in place and had already been initiated. A wider surveillance of the vectors, of course, and mosquito control activities. Development of technologies incorporated climate information into early warning systems for infectious disease predictions. And Chloe mentioned that in the previous session. Importantly also for yellow fever, as there is a vaccine. Vaccination is really a key driver, as one dose provides long life protection against the disease for most people. So for yellow fever, I know that there is a vaccine, so it's really a difference compared to other vector-borne diseases, mosquito-borne diseases. However, there is still lots to do to not reverse a century of health gain. So, for example, it would be really key to integrate this climate information in yellow fever vaccine long-term demand forecast to secure to have the appropriate amount of vaccine and the sustainable supply of vaccine. And also, importantly, partnership, cross-sector commitments from global institutions, governments, and industry will be key to increase vaccination coverage rates, to illustrate just there the power of collaborating approaches. More than 330 million people were protected against yellow fever in Africa since 2006, with almost 70% since the eye program inception in 2016. And for those that are not familiar with the eye, is that the global long-term strategy developed by WHO with core partner, many Gavi and UNICEF, to eliminate yellow fever epidemics. So I think this figure really illustrates the power of the collaborating approach and how we could boost the prevention on disease. Also, partnership will be key to support the innovation to really get prepared for future large unexpected outbreaks. Support innovation to improve diagnosis and surveillance, that would be really key for a rapid detection of outbreaks. And also to develop probably new yellow fever vaccines, no more dependent on pathogen-free eggs, that can really improve the rapidity of response, securing stockpiling, improving emergency response to be really ready to tackle these potential future large unexpected outbreaks. And maybe just to hand few key takeaways that I wanted to pass to the group. Let's not underestimate how climate and also human behavior change could impact even all well-known diseases like yellow fever. And let's get prepared. All and each actor from the global health ecosystem has really a role to play to support partnership and innovation at all levels in order to avoid modeling data to become a reality.
Annabelle, thank you so very much for this really, really important overview of forgotten diseases making a comeback, yellow fever, along with chikungunya, dengue fever, Zika, so now, but hugely dominating the news a few years ago. Really some major changes here in the global health landscape with these arboviruses and very much driven by climate change. And thank you for your reminder of all the touch points between climate and yellow fever, whether it be on the virus itself, but also on the mosquito, on the human behaviors, whether they be social or environmental changes. And very importantly, and I hope we will have some time to discuss that, really opening up the frameworks of climate impact on health to these all important cross-sector partnerships, particularly given really the climate emergency. So we'll come back to that, and thank you. That was a brilliant presentation. I'm very sorry to learn yellow fever was also called black vomit. It's not got quite the same tone to it, but very interesting. Thank you so much. And so before we turn to our discussion, I'd like to hand over to our last presenter of the session, Dr. Carlos Andres Bravo Vega. And Carlos, thank you so much for joining us at what is a very early time over in Colombia, but we're looking forward to hearing all about your work linking rainfall and temperature and snakebite.
Okay, Marianne, thank you very much for inviting me to this great conference. Are you hearing me right?
Absolutely, yeah.
Perfect. I'm going to share screen. Thank you very much for the invitation. Thank you very much for being here. My name is Carlos Bravo. I'm from the University of Los Andes in Colombia, and I'm going to talk about this work that we did that is called Disentangling Snakebite Dynamics in Colombia. How does rainfall and temperature drive the snakebite temporal patterns? So to start introducing this topic, something really important is that snakes are a really diverse animal. You can find a lot of shapes and color of different snakes, but not every snake are venomous. There are some snakes, such as this group of snakes, where you can find the boas or the anacondas, right? That doesn't have any fangs or venom glands. But on the other hand, you have these two groups of venomous snakes. Here in the tropics, you have the coral snakes from the family Elapidae, but for example, in Africa and in Asia, you have the cobras and the mambas, which are venomous. They have fangs and they have a venom gland. And also you can have the viper snakes, which you can see like the fer-de-lances, the bushmasters, the rattlesnakes, which has also fangs and also they has venom glands. So not all snakes are venomous. The most probable thing is that if you find a snake, it will be a non-venomous snake. But this tiny group of venomous snakes cause this problem, this disease, where we have an estimation of around 1,800,000 cases each year with at least 94,000 deaths. But something important is that snake bites fulfill all the requirements to be treated as a neglected tropical disease. So nowadays, the World Health Organization recognizes snake bites as an NTD, and that's the reason why I am here talking with you about snakes and venomous snakes. If we compare mortality of a snake bite with these other neglected tropical diseases, we can find that it has the first place in deaths. But something that happens with the snake bite is that normally we don't know anything about the natural history, ecology, and basic biology of venomous snakes. For example, for the sandflies in leishmaniasis, or for the water snails in schistosomiasis, or for the kissing bugs in Chagas disease, we know reproductive cycles, we know home range, we know activity patterns, but all of this data is totally unavailable for snake bite here in Latin America. Another thing that happens in snake bite that is transversal to other neglected tropical diseases is underreporting. When a person has a snake bite, it can go to a medical center where the case can be reported, or he can go to seek for traditional medicine where the case will not be reported. So as a consequence, data underestimate disease burden. The treatment of a snake bite is done by antivenom, which is produced by immunizing production animals. So you require a serpentary with veterinary care for venomous snakes. You require to have care for these production animals, mainly horses, and also you require to have a laboratory where you are going to process the blood samples of the animals to produce the antivenom. So this is something that is expensive, although antivenom is effective, it has limitations as it doesn't always reach where it is really needed. So vulnerable populations become unattended. So in Colombia, we have around 320 snake species, but only 51 species are venomous, right? But of these 51 venomous species, these two species, Bothrops asper and Bothrops atrox, which are pit vipers, cause most envenomings, right? They cause between 60 and 80% of total envenomings. In the country, we have around 5,000 yearly reported cases with at least 50 yearly reported deaths. So it's something like 1% of a case lethality caused by a snake bite if people go to medical centers and get antivenom. If we see the distribution of hospitals with the capacity to deal with a snake bite in Colombia, we can check that in several areas we have hospitals that have to cover huge areas and people will have to travel long times to that hospital. In addition, distribution became challenging, antivenom distribution became challenging. So, for example, in 2017, if we look at the severe cases, which are cases that put in risk the life of the patient, the 13% of these cases, people didn't get antivenom. And as a consequence, people are dying because, not because we don't have antivenom, but because we are not being capable to make this treatment available for vulnerable populations. So people do the most difficult part, that is arriving to a medical center from their communities, indigenous communities, rural communities. But when they arrive the medical center, they don't have antivenom and people die in these hospitals. So in this kind of problematic, the World Health Organization developed a global strategy that aims to reduce snakebite mortality and morbidity by 50% by 2030. They developed 4 strategic aims, and we are going to focus on the third aim, that is a strengthened health system. And to strengthen health system, we need to improve the information regarding epidemiology of this problematic. And to start doing this, given that we don't know so much about the biology of these venomous animals, we have to start trying to model, to mathematical modeling, these kind of diseases. So the first thing that we wanted to do is, okay, can we model a snakebite as an infectious disease? And to do this, we tried to implement the law of mass action that is widely used on infectious diseases. This law states that the incidence, the new infection, will come from contacts between susceptible and infected populations, and these contacts are going to be modeled as the multiplication of the abundance of susceptibles and the abundance of infecteds. In a snakebite, it is not an infectious disease, but we know that new cases of snake bites will be caused by contacts between susceptible population and venomous snakes. So we can state that incidence will be proportional to the multiplication between humans and venomous snakes. But doing the measurement of venomous snakes abundance in field is really difficult. So we went to Costa Rica. Why Costa Rica? Because this country has law on reporting. The Instituto Clomiro Picao is a pioneer dealing with the snake bite. So it's a perfect country to start working with mathematical modeling to test these models. And there we tested this law. So in this plot, for each municipality in Costa Rica, we know S, that is the rural population, and F, that is a proxy for venomous snake abundance that we developed in field. So we went several nights during 1 year in different places of Costa Rica to search for venomous snakes during nights, and we performed this kind of venomous snake census. And when we multiplied both variables and compared it with the reported incidence, we got this linear relationship that is telling us that the law of mass action is working on a snake bite. So now that we know that this law is working on a snake bite, let's try to apply it to understand underreporting in Colombia. In this work, first, to estimate the abundance or the proxy of venomous snakes abundance, we did niche modeling, species distribution modeling by entropy maximization for the both species that I told you that cause most envenomings in the country, right? So when we got these maps of the suitability that we called a snake niche modeling score, and we compared these results with the reported snakebite risk, we got another nice linear relationship that is telling us that the law of mass action is working, and also that SNFMS, the results of these distribution models, are a valid risk estimator. So we got this risk estimator, and to estimate underreporting, what we did is to get an accessibility score that we computed it as the average travel time in each municipality, travel time from each part of the country, and each hospital with the capacity to deal with the snake bites. So we developed by geographical information system tools this map of travel time to nearest medical center, and we mixed this data with data of poverty that we use the unsatisfied basic needs. And with all of this data, we calibrated a generalized statistical model that estimated underreporting, the percentage of cases that are not being reported in each municipality in the country. We got that the national underreporting is around 10%, 500 cases that are not reported, that it may not sound so alarming, but if we assume that these cases that are not getting reported will not receive antivenom, and we assume a mortality rate, a lethality rate of 10% in these cases, we will have that underreporting of deaths will be around 50%. So practically in terms of deaths, we are only collecting the half of the information of the total panorama in the country. These behaviors that we found here are affected by the distribution of the venomous snakes, and it depends strongly on climatic variables. So climatic change will affect snakebite dynamics, and we could not be prepared. We will have venomous snakes in areas where before we didn't have venomous snakes. We will have some areas with more abundance of venomous snakes. So the risk will increase, the cases will increase, and if today we are not able to distribute antivenom correctly, we will have problems. And then what we wanted to do is to understand how does temperature and rifle drive snakebite temporal patterns. So to perform this, we use like the framework that is that in Costa Rica, in this work, they found a relationship between rainfall and snakebite incidents, and they explain it by the reproductive phenology of venomous snakes. So if you see at the right panel, you can see that births of newborns of new venomous snakes occur close to the rainy seasons in the Pacific and in the Caribbean of Costa Rica. But although we have the same venomous snake species in Colombia than in Costa Rica, Botrops aspect, you can see that the populations that exist in Costa Rica are totally different than in Colombia. So we cannot assume that we will have the same behavior. We perform a model based on partially observed Markov processes, where we have a process model based on the law of mass action, and we develop a measurement model based on a Poisson process, and we adjust this model by using covariates of temperature, of rainfall, and of seasonality that was not driven by any climatic variable to check which variables are contributing in each part of the country. So nationally, what we found is that rainfall drives snakebite incidents, right, as you can see in the upper panel, but maximum and minimum temperature don't. And this is because we're in a tropical country where annual temperature is quite constant. For example, in temperate areas as in the North American continent or in Europe, we can expect that temperature will drive snakebite incidents. But when we start looking for different regions in the country, what we found is that regions with high rainfall, snakebite incidence is not driven by rainfall, right? We found only in this region too a seasonality on snakebite data, but this seasonality is not explained by rainfall. But if we look at these other areas of the country where we have marked dry seasons at the beginning of the year, what we can see is that rainfall drives snakebite incidents. Why do we have this complex pattern? We don't know, because we don't know information about venomous snakes. It may be caused by reproductive cycles. It may be caused by changing activity patterns, not only of venomous snakes, but also by humans and farmers. But to start like filling this knowledge gap, we create Botroperio research group, where we are trying to generate novel information about venomous snake biology. And I want to talk briefly and quick about these three people of the group. First is Jorge. Jorge is a local farmer who has helped us a lot, and we have learned together a lot about venomous snakes. So it's really beautiful to have this relationship with people from the community. And Sophie Nata, who are the first graduated persons of this group. So, for example, in Nata's thesis, what we did is to try to understand how those vipers move. And we did a technique that is based on a bobbin thread, where we attach the bobbin thread to the snake, and then we follow the thread trail that the snake leaves on while it is moving. And with this, we can estimate home range, territory, movement rates, and we want to start doing this estimation during dry seasons and rainy seasons to determine if the relationship that we saw previously is caused by an increase in activity patterns. We found that bigger snake, SBL, is the measurement of the large of the snake, and the weight is the weight of the venomous snake. We found that bigger snakes has larger home range, so they will be more prone to reach human areas, but we have to perform more research in that part. And finally, Sophie's thesis tried to understand how does this animal perceive us, and we based this research on thermoreception. Why on thermoreception? Because pit vipers are called pit vipers because they have this loreal pit that is a specialized organ that they use to see temperature. Practically, they see temperature with this kind of organ. So what we did was an experiment to determine when are these vipers having an attacking reaction of an experiment. So I'm going to show you two quick videos. I'm going to put this smaller to don't see all the video. You are still looking at the slides, right?
Yeah.
So in this case, we have this is the same individual that I'm going to show you later. This individual of venomous snake has the eyes uncovered, is totally looking around and totally with all of the perceptive organ open. And we are passing this thermal plate at 38 degrees to check what is the reaction of the snake to this plate. This was done during the day, and if you can see, the attitude of the snake is really neutral, right? Now, when we do the same experiment with the same individual of a snake during night, right? And here we did something really interesting, and is that we covered the eyes of the snake. So the snake is not looking anything. It's only using thermal information. During the night, look the difference of the behavior. First, look how the snake follows the stimuli with its head. It is using his tongue to detect like what is happening, what is that. It's really reactive, and after that, it starts attacking. Right? And look that the attacks are really directional. A snake has a really stressed pose, is not comfortable with that stimuli, and that's what happened before a snake bite. Let's remember that we are mammals. We have temperatures close to these 38 degrees. So what we can see here is that during night, these animals become more responsive, so the risk will be higher. Another important thing is that we only register attack reactions with the thermal stimuli of 38 degrees. So, final remarks. Snakebite dynamics will change with climatic change. Global warming will increase distribution areas for venomous snakes. Rainfall patterns variations due to climate change will change snakebite dynamics. We don't know how these changes are going to be because we don't understand venomous snakes, so it's time to start prioritizing studies on venomous snakes ecology. So thank you very much for being here, and I will be open to questions in the next section.
Well, thank you very much, Carlos. We learned so much in this presentation. As you say, a very complicated exercise to work on snakebite, given the differences and the range and the varieties of snakes in the first place. So thank you very much for summarizing that. And what was really interesting from your concluding slide, which covered things such as dynamics will change with climate change, but also global warming increases the distribution, and we don't know enough about venomous snakes. This really can apply to mosquitoes. It can really apply to all the various contexts where echinococcosis is appearing or re-emerging and spreading. And I think despite the very different topics we've heard through this session, those are some really common themes. So thank you to all our speakers for bringing all these issues to our attention. And I suppose in the few minutes that we do have left to wrap up this session, something that I'd like to ask all of you, and I hope that would be helpful to our audience or to anyone who is working in diseases and worried about or investigating the impact of climate change on disease dynamics, is really, based on your work, how difficult has it been to jump from the science to then this all-important policy that we're talking about, that we're hearing in the build-up to COP28, or that more generally? And this could be from your experience from your various diseases, or just more generally as a scientist. How do we make all these really strong cases, this very solid evidence base, translate into an impact in terms of climate, or just more generally in terms of mitigating the climate impact on health? And perhaps I'll just go around the speakers in order of presentations. Paige, how do you see the gaps that you've highlighted being improved?
Yeah, that's a big question. I think that there's kind of two worlds that I wind up working in, where there is identifying gaps in the research trends so that other researchers can be aware of where the gaps in the trends are. so that we can target research towards that direction. But if research doesn't inform policy, it's not putting in the work that it could. And I think that part of being a climate scientist and also working in infectious diseases, especially in the United States at the moment, is being a very effective communicator, so that you're not, as it were, muddying the waters, and making sure that your point is getting across and there's not any kind of misunderstanding. And I think we've all learned throughout the COVID pandemic that it's very important for us to know how to present our findings and our research to the public, know how to say that we were wrong and that we've changed our minds, and knowing how to be an effective science communicator is really, really important.
That's a really important point. Thank you. Adrià, over to you.
Yeah, thank you. Well, I think yours is a very hard question, because I think in the field of climate change and infectious diseases, I mean, much more research is needed first, and things are not so clear. It's some people, I think, speak a bit too boldly about the effects of climate change on infectious diseases, and you need to go case by case, regionalize the effects of climate change that are different throughout the world, and so on. So it's hard to answer how then to move to policy. But if we go just to climate change per se, where evidence is very solid, I mean, with anthropogenic forcing of the climate system, this is something that we cannot discuss. More research is needed, that's true. But even in this case where things are super solid, policy, things are not moving in the end. And so I don't know. I think probably I cannot answer. This is more for a psychologist or other people working on how to convince people. I think the works of scientists in climate change, sometimes there's much more work to do, but up to now, the work is done. We know that what causes climate change. But now it's like, how to move to action? I don't know.
Fair enough. That is the big black box. And perhaps I should have rephrased my question slightly, which is maybe what would you like to see so that your research is taken up more rapidly? But again, that could be just another big question. If you're not adding to that, I might pass on to Annabelle.
Thank you, Marianne. So just to jump into what Paige said, I think really a scientifically driven awareness is key for call to action to maybe further research, call to action also to innovation, as I tried to highlight in the presentation, and even more importantly, to cross-sector commitment and partnership. And I think things are moving. For example, on arboviruses, we see integrated initiative putting in place, like the WHO Global Arbovirus Initiative, and I think really, partnership is really key and to address and tackle this climate change impact on health. So that would be my word to your question.
Thank you. And you made that very clear in the presentation as well, all the various avenues, certainly when it comes to arboviruses, that need to be explored and strengthened. So thank you. I'll ask the same question to Carlos.
Okay, Marianne, but I agree that it is a really difficult question, because I know that as a researchers, scientists, our motivation is to try to help to enhance the response that we have to these kind of diseases, right? All of our motivation came from the basic characteristics of MTDS, that are diseases that happens to people who are really vulnerable. But generating this kind of connection with policymakers, with national health systems, sometimes it's difficult because of different factors that as scientists, we don't know how to deal with them. For example, here in Colombia, I have a nice relationship with the people from the National Health Institute that develops the antivenom, And we have tried several times to do collaborations, but bureaucracy to perform all like the permits, and we need like how do you say that? Like formal association of a group with a formal research question, and it's a process that can take like 1 or 2 years. So when you, as a scientist, do look like at the resources that you have to invest only to get the permit for performing this collaboration versus the investment that you have to do to produce novel results, it's really difficult to choose between them, and it's really difficult like to break that barrier that it must be done. But I think that we need like more help from the health institutes, from the national policymakers, to make it easier for us to perform these contacts. We are scientists, we are not lawyers, we are not social communicators. They have to understand that we are not good doing this kind of tasks. So I think that they should help us most to break this barrier.
Fantastic. That's a really good point. Thank you. All 4 points were really important. There is quite a specific question here about snake behaviors from Tahani Alias. What about snake behaviors? We know that this venom is in the balance of the snake, so we need to know the defensive mechanism. That's quite specific to you, Carlos. I don't know if you want to reply in the chat.
Okay. If you want, I can reply in the chat while we continue with the discussion, or I can answer it.
Yeah, sure. Go ahead.
Okay. So, Tahani, this is something really interesting because we, as I told you, we don't know much enough about this kind of ecological traits of venomous snakes. We know that, as you say, the venom is the balance. It's like the snake develop the venom first to ensure their prey, but secondly, as a defensive mechanism. But the mechanisms or the information that the snake gather to determine that somebody is is threatening the animal and to take the decision to attack, we still don't know that. So this is something that it would be really interesting, because if we understand that, we can generate strategies that break this kind of behavior of these snakes, and we can start preventing snake bites.
Wonderful, and certainly your videos were fascinating, and we learned a lot about snakes. Thank you, Carlos. And just very briefly, we're coming to the end of our session, and there's never enough time. But we've heard very different insights and inputs, and just turning to the future, we know climate change is going to be a huge challenge, and I was just wondering if you could give us a few pointers before we close the session about what do you feel would be the biggest challenge in terms of climate that will be posed to the disease that you were talking to us about today. And I'll go again in order of the speakers. Paige, you obviously covered a wide range of diseases, but what were your sort of insights in terms of what you will see as the biggest challenge to having a more balanced research moving forward?
Yeah, I think sometimes it is important to address climate change as the global threat that it is, and have research be a little bit more mission oriented, in the sense that we need to be filling holes and not doing research that's trendy or research only on one area or in one place. Really taking a step back and looking at the distribution of research as it is, making sure we're being equitable in how we're devoting research funding and research interest. Specifically, some of the systems that I study that are very much animal focused, sometimes it's hard to convince people that we need as much funding as we're asking for. But I think in not devoting effort towards those wildlife-borne pathogens, or those pathogens that are carried by less well-known systems, we're really missing a step, and making sure that we are being as equitable as possible and devoting our research focus is an important thing for us to do as a field. So really communicating with other scientists and working with other researchers to identify what the field needs at that moment.
Thank you so much. Adrià.
Well, the first problem that we had with this disease and with others is the lack of data. This is a super serious challenge, because if I don't have data, I cannot say anything or do anything. No, and for this case, the massive underreporting and the fact it strikes local isolated communities in Panamazonia, this creates, well, lots of issues. Regardless of that, I didn't have time to explain, but maybe it's worth now that you asked me. Like, this is a disease which is extremely easy to halt transmission. I didn't explain the full cycle because we didn't have time, but as long as hunters burned the viscera of vacas, and normally they don't eat the viscera, they eat the meat, but they give the viscera to the dogs, and this is the problem. And if they just burned this or did something with that, the disease would be, I mean, there would be no spillover of the disease. And then it's a behavioral thing that could be changed super fast. It's easy to say. Then you have to inform communities, change all these, and so on. But yeah, it's frustrating when you see this. And then I'm studying the effects of climate change, and this is relevant, but this could be stopped much easier. But again, our results do show concerning trends regarding hunting-related diseases and how climate might affect hunting patterns, not only for this disease, but others. So, well, this will be challenging in the future, for sure. But if to summarize, I would say that, yeah, lack of data of this disease, of hunting practices, and so on. Yeah.
Wonderful. Thank you. Lots of issues to keep an eye on. Annabelle, vector-borne diseases and climate, lots of troubled waters ahead, clearly.
Yeah. Thanks, Marianne. So I think really regarding yellow fever, the main challenge, and I try to highlight that, is that, I mean, it's quite an old story. We have already some tools, we have already vaccine, it's already pretty well known, but I think the link to climate change is not enough highlighted, and there is not enough awareness for yellow fever. I mean, because there is a dengue, of course, there is a chikungunya, there is Zika that is always mentioned, but you know, we should not forget, that's a message for today, and that's, I think, a big challenge. We should not forget yellow fever, and we should not forget to get prepared for this potential unexpected outbreak, because having some tools is not saying that you have the right tool to be prepared for that, and be ready to use them appropriately to be able to tackle this risk. So yeah, I think that's the main challenge for this disease in the context of climate change.
Wonderful. Thank you so much, Annabelle. And Carlos, for the concluding words of this session.
Okay, I think that not only for snakebite, but for most neglected tropical diseases, as my colleagues had said, it is important to break the way as we communicate our results. It is really important also to start gathering better quality data to understand deeply these factors that are causing that climate change affects these kind of diseases. So I think that given that we are working on these specific diseases, a multidisciplinary approach is really, really important. And to perform that correctly, it is really important to start making connections with different people, such as we are doing here, to start making worldwide collaboration that allows us not only to have like local perception of the diseases, but to understand them globally and also by different research scopes.
Wonderful. Well, to our panelists, thank you so much for so much food for thought, coming from very different perspectives and angles. And we've really learned a lot with you, and it's been an extremely enjoyable session. So a very big thank you. Big thank you to our audience who joined in as well. Our next session is about to start in a few minutes, and we'll be looking at the impact of the healthcare sector on the climate, but also what can healthcare stakeholders do given their huge impact on climate, to mitigate the climate impact on health. So really fascinating session coming up ahead. Please do join us. I'm just sharing the link in the chat if you want to hop on to that next session. But from us at ISNTD, another huge thank you to our 4 panelists, and we'll be more than happy and very keen to keep in touch and see what the next steps are from every side. Thank you so much.
Thank you.
Thank you, Marianne.
Thank you very much.
Thank you so much.